And we are recording. Let's see. Key is not here. Boggs is here. Bonner is not here. Clay is here. Dawson is not here. Ellis, yep, is here. Paula is online. Green is not here. Harrison's here. Vincent is here. James is here. Kesterson is here. King is online. Lewis is here. Linda Q is not here. Morrison is here. Grace is here. Ward is not here. Wilcox is here. Wilder is online. Parks is here. And let's see, Dawson is here. Anybody I didn't call? We're counting for at this point. 8 o'clock. Let's see. Go check online really quickly. Yep, that's it. All right. Let's slowly get started. Maybe some more people show up. So let me think. Reminders. There was no pre-lab due on Friday, this past Friday, because this week we're going to meet like we usually do, same room, same time. But instead of doing an experiment, I'm going to give you the pre-lab lecture this Wednesday. And then I'm also going to teach you how to write a lab report this Wednesday. That way, next Wednesday when we show up, we just do the experiments. We want you to do a pre-lab lecture. We can just jump right into it. It's one of those, these experiments take a while. There's a lot of waiting. So that's another good reason to just jump right into it instead of having to do a pre-lab lecture. So any questions about how lab's going to work for the next couple of weeks? All right. And again, you know, your pre-lab for next week is due this Friday. So I recommend getting started on it early, especially if you could. It would be good to get it done before, you don't have to, if you were to get it done before we meet on Wednesday. That way you'll know what questions to ask me when we have our meeting. Any questions about that? All right. Any questions about anything in general? Don't forget to get some independent work done. So far off the top of my head, only 3 of you have turned anything in. And 2 of you seem to be having a hard time, but trust me, you're not, because no one else is turning anything in. So for those of you who are having a hard time, keep in mind that you're well ahead of everybody else, except one other person. Anyway, any questions before we jump in?
How long would the independent work have to be for it to be 100, like just for all of one paper?
Good question. So, and I'm not saying this, I'm going to answer the question, but also if you forget what I'm about to tell you, don't forget you can look at this in the syllabus. But you have all semester to get 100 points. So whether you write one huge paper that's perfect and 10 pages long and get 0 points deducted because it's 10 points per page, then you could write one paper, or you could write 100 small papers. It doesn't matter. Just keep turning things in until you hit 100 points.
It's 10 points per full page?
Yes, 10 points per full page if it's perfectly written. For those of you who have turned stuff in, you know what I mean by perfectly written, right? I will find things. I want to challenge you, right? So I'm going to look through your paper and say, hey, what about this? So make sure, it's not grammatically that I'm going to be challenging you on, it's the content. As some people will know, like, hey, I read your paper, I read your source, and this information in your paper was not in your source. So please reply to me and correct it. It'll be things like that. So if you're good at writing, I say go for it, write some big papers. But if you're not good at writing, start turning in small papers. Again, just think of a question and answer it. And if it really is just a question and answer, you could just send it to me via email instead of writing a paper. You could say, again, using the same example I've used before. I was wondering why armpits smell. I looked it up, and according to this source, it's because of this reason, right? If it's just a couple sentences, just email me. That's 1 point if you correctly answer a biology question. If you want to write a paper about it, then go ahead. And there is no minimum or maximum length of the paper. So I would start turning them in. We're 5 weeks? We're about halfway through the semester. So in theory, you should have about 50 points by now, right? In theory. And if you need ideas, let me know. I'm working on, so one of the things I'm working on the side is like a list of ideas and some worksheets that you can use to help you write papers. You'll see what I mean. I'll share them once I get them all done. But anyway, any other questions? That was a good question. All right, and remember, it's 100 points. So 100 points out of 1,000 points, that's a full letter grade. You say you're bad at testing or things like that, then this is supposed to help you with that. This is completely in your control. Just write 100 points worth of work. Anyway, back to cellular respiration. Actually, first thing, let's do the attendance word. Let's say breakdown is the first attendance word. Right there, breakdown. Like the Tom Petty song. Anyway, we are talking about cellular respiration, which is broken down into 3 stages: glycolysis, citric acid cycle, and the electron transport chain. I was telling you for most of the exam, for this portion of the exam, it'll be me describing something, telling you which stage it happens in, right? So I might say which stage produces ATP, which is all of them. Or I might say which stage uses glucose, which is glycolysis. Which stage produces pyruvate, glycolysis. So that's how the questions are going to go. We finished talking about glycolysis. In the end of glycolysis, right, the end product of glycolysis is pyruvic acid, aka pyruvate. And we talked about how it has to go through this whole process before it can even enter the citric acid cycle. And we talked about all this stuff that's on the screen, and I told you, technically, this is kind of the in-between zone between glycolysis and the citric acid cycle. As far as you're concerned, as far as the exam is concerned, all this is considered the citric acid cycle. So again, before I move forward, some test questions that are on this page are, which of the 3 stages uses pyruvic acid or pyruvate? Not produces it, but uses it. That would be the citric acid cycle. Which of the 3 stages is associated with acetic acid? That would be the citric acid cycle, right? Citric acid cycle. Which of the 3 stages is associated with acetyl-CoA? Citric acid cycle. Which of the 3 stages produces carbon dioxide? Citric acid cycle. Which of the 3 stages strips away high energy electrons? Citric acid cycle and glycolysis, right? So a lot of test questions on this one picture alone. So that's where we left off. Any questions before we finally jump into the citric acid cycle? Yes, ma'am.
Glycolysis is listed to have
Yep. Yeah.
So it's all 3?
No. Like I was saying, which of the 3 stages strips away high energy electrons? Citric acid cycle and glycolysis.
Oh, I thought you said citric acid and electron transport.
Oh, no.
Just kidding.
And who knows? I may have said that, and if I did, thank you for correcting me. Definitely not, because, matter of fact, while we're saying that, let's back up to the overview. The first 2 stages strip away high energy electrons, and the 3rd stage uses those high energy electrons. In a sense, the first 2 stages are here to support the 3rd stage. And remember, the 3rd stage makes the most ATP. So when you think about it, it's all about that 3rd stage, the electron transport chain. Any other questions?
Can we check the pH level on these acids, or would that be?
We can't. I mean, we in this room can't, but that'd be something to look up for. Look it up if you want. I'm sorry. Yes, we can't check, but you could look it up for independent work. What's the pH of acetic acid, for example? What's the pH of citric acid? Yeah, that'd be something to look up for independent work. Good thinking. So again, this is the end. This is the middle zone between glycolysis on this side, citric acid cycle on this side. So now that we have this acetyl-CoA, that thing is now able to go into the citric acid cycle. So let's actually talk about the actual citric acid cycle. And in this process, we have 2 carbon dioxide molecules that are produced, but you don't need to know the number. Just need to know when you get the test question, in which of the 3 stages is carbon dioxide produced? And the answer is citric acid cycle. And again, I've said this before, but there's a little preview. When we start talking about photosynthesis, then the question is going to be which of the 2 stages uses carbon dioxide. And the good answer, the good news for you is it is a different type of cycle. So again, when you hear anything about carbon dioxide in this exam, you should think of cycle, whether it be the citric acid cycle or the one I'm going to teach you when we talk about photosynthesis. And again, like I said, some ATP is produced directly, just a little bit, not much. And again, that's another test question. Which of the 3 stages produces ATP? All of them, but the first 2 only produce a little bit. But again, you should see a common theme at this point. much more energy is captured as high energy electrons. And remember, when you see these, any molecule that looks like this, you should think high energy electron. And I told you when I introduced you to NADH, I said, all right, there's going to be two more, and you don't necessarily need to differentiate between the two, but when you see them, you should think high energy electrons. So here's the other one, FADH2. You can see how that molecule just looks the same, right? It is slightly different, but as far as we're concerned in this class, these things are the same thing. All they do is strip away high energy electrons. And remember, that's what the first two stages are all about, stripping away those high energy electrons so that the electron transport chain can use them, because that produces the most ATP. And then finally, the textbook points out that all carbon atoms that enter the cycle as fuel, aka that acetyl-CoA, are accounted for as carbon dioxide and a 4-carbon acceptor molecule, which is recycled. So I would put an X to this bullet point, and I only included it because your textbook did, and it's kind of a good segue into this next picture. So again, what we have here is acidic acid cycle meets up with this 4-carbon acceptor molecule. Not that you need to know the numbers, but I'm just explaining how it goes. So 4 plus 2, that comes together, and you have 6, right? So you have the 6-carbon citric acid cycle. We break that down because, again, when you break down molecules, you're releasing energy, right? So in this case, when we break that down and release energy, we're making ATP from that energy that's being released. And more importantly, we're stripping away high energy electrons when we're breaking down the citric acid cycle. And of course, as a side product, we're also producing carbon dioxide. So again, we start off with a 6-carbon molecule. We lose 2 of those carbons to carbon dioxide. Obviously, 6 minus 2 gives you 4. And then we're back where we started, right? The cycle continues. And you could look this up. This is a very, very, very simplified version of how this actually happens. So this acceptor molecule actually has a name that you could look up for independent work if you want. There's a lot more steps in here that are not being shown in this diagram. So you could look those up. What are the different steps that are not being shown? What are the different molecules? Because again, if there's more steps to get us between here and here, there's also more molecules to get us from here to here. And they all have different names. And they also have different enzymes. Each enzyme for each step that is not shown has its own enzyme. So you can look all that up for independent work if you want. Anyway, for test questions on this slide, once again, I know this can sound redundant, but hopefully it'll help you do well on the exam. Which of the 3 stages is associated with acidic acid? Acidic acid, right? Citric acid cycle. Which of the 3 stages is associated with citric acid? Obviously the citric acid cycle. Which of the 3 stages produces carbon dioxide? Citric acid cycle. Which of the 3 stages produces ATP? All 3. The first 2 only produce a little bit. Which of the 3 stages strips away high energy electrons? And that would be glycolysis and the citric acid cycle. That's it. I'm not even going to ask you how the cycle works. I'm not going to say, you don't need to remember the fact that this 4-carbon acceptor molecule meets up with this 2-carbon acidic acid molecule, and that gives us the 6-carbon citric acid molecule. I'm not going to ask you all that. So any questions about this picture?
What is the type of energy that's released from this?
This is all
Is it
chemical energy.
chemical, electrical?
You could call it that. Yeah, you could, because we're dealing with electrons. But it's not electrical energy like we might think of as, electrons running through a circuit, running electronics. But yeah, it is electrons. It's funny you should mention that, because that's the next point I'm about to get to. Because the next thing we're going to talk about is the 3rd stage, which is electron transport. This is what I call it. Your book calls it oxidative phosphorylation. But again, I just call it electron transport or electron transport chain, or ETC. So it's a great thing that you asked that question, because you really, in a sense, can think about this as an electric circuit. So I'm sure you guys have either jumped a car before or seen it done. Electrons have to come from somewhere and they have to go somewhere, right? So this is another thing that you're going to need to know for the exam. Separate from the stuff I've been telling you, because remember, I've been saying, generally speaking, it's in which stage does this happen, and which stage does that happen. Now I'm telling you there's one more thing that you need to know, and that's basically the story of the electrons. Where do the electrons come from? Where do the electrons go? Right? Because in cellular respiration, these electrons have a fall and cascade. Because notice every time I was talking about electrons before, I don't just say electrons, I say high energy electrons, right? They've been energized. We'll talk about that in a different chapter. But yeah, those are energized electrons, and as they go down the stage, so to speak, or down the stairs, so to speak, they're releasing their energy. And that energy has been put to use, right? And so far, we've used that energy to make a little bit of ATP, right? That's what we've been using it so far. And again, we've been stripping away those high energy electrons that have to go to the electron transport chain. So this brings me to my point. As in any electric circuit, electrons have to start somewhere and they have to go somewhere. So let's talk about where they start and where they go. Actually, before that, really quickly, just as a reminder, this is happening within the mitochondria, the electron transport chain, and we'll talk about why that's important. So the citric acid cycle also happens in the mitochondria, and you do need to know that for the exam. But for this one, the next one's a little bit more important. So it just so happens the citric acid cycle takes place in the mitochondria. The fact that the electron transport chain happens in the mitochondria is actually important. It wouldn't happen anywhere else, and I'll explain why. But first, let's take another step back and talk about these electrons. The overall effect of the electron transport is a downward trip for electrons. So again, like I keep saying, they're high energy, right? And you can look up what that means if you want. It might be confusing to you if you're not much into physics. But yeah, these are high energy electrons. As they go down the stairs, so to speak, they're releasing that electron. But here's what you need to know for the exam. Where do the electrons come from? Glucose. That will be test questions. Because this is all about electrons. It's all about ATP. We're trying to make ATP. Yes, the first two stages make a little bit of ATP. Who cares? So the third stage is making the most of it, right? And how does it do it? It uses electrons. So this whole thing, in my opinion, is all about electrons. So where are the electrons coming from? They're coming from the glucose. Now I'm going to skip the middle two bullet points for a purpose. And let's just get straight to the last. Where do they end up? Finally, where do the electrons come from glucose? Where do they end? They end at the oxygen, and that makes water. So there will be a test question. Now, I did include these two middle bullet points because, yes, technically that's what's getting them from glucose to the oxygen, right? We've got the NAD+, FADH, or FAD plus, right? So they are getting transported. That's those two middle bullet points. But for the exam, what's important is you know that they came from the glucose and they end up with the oxygen. And when the oxygen accepts them, that forms water. You need to know that for the exam. And if you can memorize that, that'll help you with photosynthesis, because there'll be a similar question for photosynthesis. Because in many ways, photosynthesis is literally the opposite of respiration. So in photosynthesis, the question is going to be, well, it'll be the same question. Where do the electrons come from and where do they go? In photosynthesis, the electrons come from water, which produces oxygen, and those electrons are eventually incorporated into a molecule of glucose, right? So if you can memorize this one, then when we get to photosynthesis, it should help you. Just remember, it's the opposite. So yes, remember, electrons come from glucose. Oxygen is the final acceptor, and when oxygen accepts it, that forms water. Are there any questions about that slide before I show you a graphic of what's on this slide?
Does the water that is formed from oxygen there, does it get the hydrogen from the glucose?
No, it's just gets it from, what's the word I'm looking for? Just inside the mitochondria. I can show you right there. So there's just hydrogen and protons, technically. So that H plus is technically a proton, oddly enough. It says it's an H plus, it says it's an hydrogen, and technically it is. But think about it. Hydrogen, not that you needed to remember, but remember hydrogen's atomic number is 1. So it has 1 proton, 1 electron. So if it loses an electron, it's just a proton. Yeah, that's where it comes from. Not that you need to know that. And once again, I'm glad you mentioned that because I can now put an X through this. I'm not going to ask you where the hydrogen came from. I'm not worried about these numbers either. So forget about the numbers. Again, what you need to know is the electrons came from glucose. I mean, technically from the food, because obviously we break down things other than glucose, and we'll talk about that later. But for the exam, the electrons come from glucose. They go through the whole process, releasing energy in the process. Oxygen eventually accepts them, and that produces water. That's what you need to know for the exam. You don't need to know that technically it's 2 electrons. For every 2 electrons, we have 1 molecule of water. You don't need to know that. And again, this is a very oversimplified version of how it happens, but it's a good base foundation for your knowledge. Any questions about this slide? All right, so the next word for attendance will be water. And also, that picture is not from your textbook. So if you're studying, you're trying to like figure out where I was in the textbook when this portion of the PowerPoint. Keep that in mind. So again, like I said, glycolysis happens outside of mitochondria, and the other two stages happen inside the mitochondria. As far as we're concerned, the fact that citric acid cycle happens inside the mitochondria, that's pretty trivial. It's not very important. But the fact that this happens inside the mitochondria is important, so let's talk about why. It's because there are proteins built into the inner membrane of the mitochondria. Right? So the proteins, the electron transport chain proteins, the proteins that are involved in all the stuff we're talking about, they are built into the inner membrane. And you'll have a much better picture on the next slide. But imagine that's a mitochondria. There's your outer membrane. There's your inner membrane. It looks a lot different than that because the inner membrane should have a lot more waves. But we'll get to that in a second. Yeah, like I said right here, it's highly folded, like in this picture. But yeah, there's a bunch of, I'll just draw little dots, right? Just a bunch of protons built into that inner membrane. But really quickly, forget that picture. Let's look at this picture. You can see the outside is nice and smooth, and the inside is really folded. You guys can see that, right, in that picture? Now imagine if you had like a big garbage bag, like, maybe they kind of fit there, it's a big garbage bag, and then the Kroger bag. Now imagine if you stuffed the big bag inside the Kroger bag, right? It'll be all crumbled up, right? Because it's big and you're crumbling it up into something smaller. Does that make sense? That's what's happening here. And the reason is because, think about it, again, the proteins that are doing the job are embedded in this inner membrane. You can't really see it in this picture, but they're embedded in the inner membrane. So the bigger the inner membrane is, the more surface area it has, the more proteins you have that are doing the job, therefore, the more ATP you can make. So that's why we have this structure. This is a good example of structure and function, right? This is why the mitochondria has this shape. Or I guess you could say the reason it has this shape, therefore it is very effective at making ATP. Anyway, any questions about this so far? All right, so again, keep in mind we have an inner membrane, which we see here, and we have this outer membrane, which means we have this area in between the membranes. And I guess we could go back and look at my horrible picture. But yeah, so right here's the inner membrane here, the outer membrane here. So this is the outside of the mitochondria right here. This is the inside of the mitochondria right here. And this is kind of the in-between zone, right? But that in-between zone is actually really important. Let's talk about why. First of all, let's talk about how a dam works really quickly, and then we'll talk about that. Think about how, I don't know if you guys know how hydropower works, but if you can imagine here, this is the dam, right? So we have a lake or something on that side where the water is building up against the dam. This is where the water comes through. And when you let the water through, once you open those gates, the water just naturally flows, right? That's what gravity does. Water will flow downhill. And when that happens, it turns the turbine, and that produces electricity. But the same thing kind of happens here, except it's not water. Here, and this is what you need to know for the exam, what we're doing when these electrons, when we take these high-energy electrons, we put them through the electron transport chain. As they lose that energy is used to pump hydrogen ions against their concentration gradient. This is why that chapter about active diffusion or active transport was so important, right? Because here we need energy because we're pushing something against this concentration gradient. And we're getting that energy from those high energy electrons that are going through the electron transport chain. So again, they release that energy. And I've said this since we started. I keep saying we release that energy and that energy is used to produce ATP. Now I'm finally telling you how that happens. Because when those electrons lose that energy, we're pumping hydrogen ions against their concentration gradient, which would kind of be like pumping water back behind that dam. And then we let it through, right? Just like we do with the hydrogen ions. So again, going back to my horrible picture here. Whoops, where are we at? There we go. We're pumping hydrogen ions from the inside here into here, right? We keep pumping it up. They don't have anywhere to go. So eventually, you're just going to have a bunch of hydrogen ions in here. And that looks horrible. A bunch of hydrogen ions in between the membranes. And what are they going to want to do? They're going to want to diffuse from an area of high concentration to an area of low concentration. And they do. And they diffuse back down. And when that happens, that's where we make our ATP. I'll explain that here in a second. But are there any questions so far? Again, the purpose of these high energy electrons, as they're releasing that energy, is to pump hydrogen ions against their concentration gradient. Of course, I won't get that specific in the exam. The exam questions on here would be, which of the three stages pumps hydrogen ions against the concentration gradient? The answer is electron transport chain. Which of the three stages lets hydrogen ions go back down their concentration gradient? And the answer is electron transport chain. Basically, if you think about hydrogen ions, you should think electron transport chain. Anyway, any questions about that? We're going to talk a little bit more about it. All right, so again, as you guys may or may not know, I kind of described it here. With hydropower, as the water is rushing downhill, right, just as gravity's pulling it downhill, that then turns a turbine, which allows us to produce electricity. And the same thing kind of happens in our cells. Except we don't have turbines that produce electricity. What we have is an enzyme that you need to know the name of, ATP synthase. That literally does turn. It has a part that turns, and that produces ATP. So again, this whole process, the whole process of all of cellular respiration, stripping away high energy electrons in the first two stages. Then those high energy electrons come here to the third stage, and they go through protein to protein to protein. And as they do that, they're releasing their energy. That energy is used to pump hydrogen ions against the concentration gradient, like water on the other side of a dam, and then we let it through. And when we do let it through, that turns this little thing on the ATP synthase, and that produces ATP. And all this will probably make more sense on the next slide when I show you a picture. But again, for the exam, that is definitely a test question. This is the only of the three stages where I told you you need to know the name of an enzyme. Because I mentioned enzymes on all the other stages, and this is the only one where you need to know the name of it. So which of the three stages is associated with ATP synthase? Electron transport chain. And hopefully, again, you can remember that the electron transport chain is the stage that produces the most ATP. So any questions about this before I show you a visualization of this? All right, this picture's not from your textbook, this is from the old textbook. So if you're studying and you like this picture and it'll help you study, then you want to come back to these PowerPoints to look at it. So here we go, here's the inner membrane. Here, what you can't see is the outer membrane. So this is the area in between the membranes, right? So again, these high energy electrons that we stripped away in the first two stages are brought into the mitochondria, and they go from protein to protein to protein. As they're doing that, that's why they kind of draw it at a downward angle, because that's supposed to signify that it's losing its energy, right? We start high energy and we get lower and lower because those electrons are releasing their energy. That energy is used to pump hydrogen ions against their concentration gradient. So then you just have a bunch of hydrogen ions stuck in between the two, the inner membrane and the outer membrane, and they just want to do what they naturally do, which is diffuse back down their concentration gradient, right? So it doesn't take any energy to get them to come down, right? It takes energy to pump them against their concentration gradient. It doesn't take any energy to get them to come down, right? It just happens naturally, and that's where we make our ATP. You can also notice too, and I'm not going to get this specific on the exam. Remember, like I told you, as far as we're concerned, NADH and FADH2 are the same thing, right? I said whenever you see anything like that, you should think, oh yeah, that's the electron, that's something that's carrying electrons. But you can see here, technically there is a difference, right? So technically, the NADH is bringing electrons to the first protein, and FADH2 is bringing them to the second protein, but we're not going to get that into that much detail in the exam. Again, all you need to know is when you see any molecule like this, and there's a third one that's used for photosynthesis, if it looks like that, you should think, yep, that's the electron, that's the thing carrying the electrons. And yeah, there are technically 3 proteins in this process. You can look that up if you want. What are the names of those proteins? But again, if you remember, let me do one more dumb drawing. Remember way back when we talked about membranes and we talked about facilitated diffusion and active transport, right? There's the membrane, cell membrane, not even mitochondria. Remember we said that there's proteins that allow things to come in and out of the cell. Same thing with the mitochondria. So that's what those are, proteins that are allowing hydrogen ions to go up and out of their up their concentration gradient. So any questions about this slide? And again, remember, This is not from your textbook, so if you want to study this picture, you're going to need to download the PowerPoint.
What are the membranes made out of?
Same thing for the cell membranes, which is a great question. I did kind of remind you, you guys need to know this. Phospholipid bilayer. Remember when we talked about it in chapter 2, we talked about lipids, and that was one of the categories of lipids we talked about was the phospholipid. And then in chapter 3, when we talked about how cells work, we talked about the membrane. And yeah, it's a phospholipid bilayer. Good question.
So would the membrane cause resistance for the negative charges?
With the positive charges, yeah. Which is why the hydrogen ions can't just diffuse in and out, right? They have to go through these proteins, which is good, because without that, if these hydrogen ions could just move in and out without the help of a protein, then we wouldn't need ATP synthase. Then we wouldn't have ATP, therefore life as we know it wouldn't exist. Like bacterial life doesn't do this. Eukaryotic, which is us, it does do this, right? So we could probably have, bacteria on Earth, but without this, life as we know it wouldn't exist. The fact that hydrogen ions cannot travel willy-nilly through the membrane is why we can have ATP. And without ATP, we don't have life as we know it. Great question. Anything else? All right, so here's another number you need to know. And I mentioned it before, but for every molecule of glucose, we produce about 32 ATP. If you look it up in a different source, like maybe even your textbook, because I don't remember if this is from your textbook or from the old textbook, but the other number that you often see might be 38. But the good news is it's a multiple choice question, and I'm not going to try to trick you. So anything in the 30s will be the correct answer for that. And again, I'm not big on numbers, but that is one of those questions you will get. How many ATP are produced per molecule of glucose? And if you take notes, you just put 30s, right? Anything in the 30s will be good. And this is, again, I don't make these names, but I find it. This is a good independent work topic if you want to look into it. Cellular respiration requires ATP, because if you remember way back we talked about the first stage was glycolysis. And the first phase of the first stage was energy investment, right? So to start the whole thing off, we needed ATP, which brings up that great question. If we need ATP to do this, then where did the first ATP come from? You can look it up for independent work if you want. And again, you could just make a quick answer. Like I looked it up, and according to this source, this is the answer. And they give me like one sentence. You just email that to me. You don't need to make it a Word document or a Google document. Just send me the answer. You get 1 point. If you want to make a paper out of it, which you probably could in that situation, because it's probably a pretty complicated answer, then you could do that too. Anyway, any questions about this before I show you a picture of the big picture? A picture of the big picture. Kind of a review of everything we've talked about. So again, we start off with glucose. We break it down in half into pyruvate, aka pyruvic acid. That thing is then going to go in the citric acid cycle, but it has to go through some changes first, right? We change that into acetyl-CoA. Acetyl-CoA then goes into the citric acid cycle. This whole process of changing the glucose to pyruvate to the acetyl-CoA, we're breaking it down, we're stripping away high energy electrons, which is the most important part. And even though it's not on this slide, we're also producing a little bit of carbon dioxide as a byproduct. All 3 stages are producing a little bit of ATP, and the big one is at the end, the electron transport chain, which produces about 28 ATP. And when you add them all up together, that's about 32 ATP. So once again, really quickly, there's a lot of test questions on here. Which of the 3 stages uses glucose? Glycolysis. Which of the 3 stages produces pyruvate? Glycolysis. Which of the 3 stages uses pyruvate? Citric acid cycle, which that's a little bit hard to see in this graphic. Which of the 3 stages is associated with acetyl-CoA? Citric acid cycle. Which of the 3 stages strips away high energy electrons? Glycolysis and citric acid cycle. Which of the 3 stages uses those high energy electrons? That's electron transport chain. Which of the 3 stages happens outside of the mitochondria? Glycolysis. Which of the 3 stages produces ATP? All of them. And which of the 3 stages produces the most ATP? Electron transport chain. And what is the name of the enzyme that produces the ATP in the electron transport chain, ATP synthase. A lot of test questions right there. Oh yeah, and how many ATP are produced? About 32, in the 30s. Whew. Goodness. Anyway, any questions about this slide? Or any questions about cellular respiration in general? Because now we're done talking about cellular respiration in general. Yes, ma'am.
It's technically not a question about this, but it is. So if I were gonna do an independent work about the citric acid cycle, because you said all that extra stuff that it does, that's not here. Would you ever accept an independent work in video form? Does it have to be written?
Yeah, we'd have to talk about it though, because I'd have to figure out the grading rubric. But yeah, matter of fact, that'd be really good. Not that this was a requirement, but if you were some kind of major where you wanted to be a news anchor or something like that, right? You could do it in the style of somebody telling, like it's a news article, or you could do it in the style of somebody who's an influencer. For some reason they thought that was entertaining and wanted to put it out that way. Yeah, I think that would be very creative and a great idea. But again, we'd have to figure out the grading. But if you're making a video, I don't know. To me, it seems like you might want to write it down anyway. So if you can write it down, I can base it off of that, maybe. But not that you have to. But anyway, yeah, we can talk about that.
I've just seen that whole thing, and it's just something I would not rather.
Okay. Actually, when you see it written, like if you were to look it up, I won't say simple, but it is at least straightforward. Because even though it looks like it's a cycle, when you see it in writing, it's this thing goes through this enzyme and becomes this thing. Then it goes through this enzyme and becomes this thing. And then it goes through this enzyme and becomes this thing. And then it goes through this enzyme. It's very straightforward. It sucks as a biology undergraduate student because you have to memorize every single step and every single name and to be able to draw every single molecule. But anyway, yeah, good question. And yes, we can talk about that later. And earlier we were talking about writing songs. You could write songs or art, do all kinds of different things. But yes, when it comes to something like that, anything that's not in writing, we need to meet first so we can discuss the grading. Any other questions? All right, so that's it for citric acid cycle. Now, remember, citric acid cycle is basically, again, this is what happens in the presence of oxygen. Because oxygen is the thing that ultimately accepts those electrons. So let's talk about what happens when you don't have oxygen, and that is called fermentation. So here are the learning objectives of this section. By the end, you'll be able to discuss the fundamental difference between anaerobic cellular respiration and fermentation. And as far as we're concerned, I would say basically those two things are the same thing. As far as we're concerned, those are the same thing. But we'll talk about that here in a second. You should be able to describe the type of fermentation that readily occurs in animal cells and the conditions that initiate that fermentation. So here we go. Let's talk about that. So again, what's happening here is basically what happens without oxygen, right? So cells, we're talking about our cells, like eukaryotic cells. Eukaryotic cells can work a very short period of time without oxygen. This is why when you hold your breath, you don't die right away, right? Hold your breath for a little bit. Or more importantly, here's a better example. When you go running, like if you're sprinting or doing something where, maybe you're out of breath, that's because you're using more oxygen than you're bringing in, right? You're breathing in heavy because your body is using more oxygen than you're giving it. And have you ever done that? You felt like burn in your legs or something? Does that ever happen to anybody? You're like running, sprinting, and your muscles are burning? That's because of this. Well, among other reasons. It's because of fermentation. So what's happening is your cells are running without oxygen. And we're going to talk about where that burn comes from here in a second. But yes, you do need to know this for the exams. What is it called when cells produce ATP without oxygen? It's called fermentation. Also, you need to know the term anaerobic, but that's kind of in the name, right? So without, and aerobic, so without oxygen. Any questions so far? Man, that's an old name. Anyway, here's a number you do not need to know, but you could look it up for independent work. I don't even know this, but I would assume, first of all, let me read what the bullet point says. After functioning anaerobically or without oxygen for about 15 seconds, muscle cells generate ATP by fermentation. So then that begs the question, what about other cells? That's just muscle cells. What about all the other types of cells in your body? You can look that up if you want for independent work. Also, as always, for independent work, you can just look up a different source. So I'm telling you it's about 15 seconds. Look it up, find another source. Does another source also say about 15 seconds, or does it say exactly 17 seconds, or nearly 20 seconds, whatever it may be? You can look that up. Anyway, because we're doing this without oxygen, fermentation relies only on glycolysis. That's another thing you would need to know for the exam. If we're talking about fermentation, we're not talking about all three stages of the cellular respiration. We're only talking about glycolysis. And as we know from our discussion about glycolysis, it only produces a little bit of ATP. This is why, even anaerobically, eventually you will die, because that's not enough ATP to support our bodies. So why only glycolysis? Why not citric acid cycle? Why not electron transport chain? And this is the answer why. Glycolysis does not require oxygen, technically. When you think about it, right? In glycolysis, all we were doing was stripping away high energy electrons, in the process producing a little bit of ATP. Right? But there was no oxygen directly there. Wish you could say the same thing for citric acid cycle 2, but we won't get into that. So yeah, that's the reason why glycolysis is used for fermentation, but let's talk about that a little bit more. What we need for this process is NAD+. Not that that's going to be a test question, but to remind you, when it has the plus, that's that molecule that's carrying electrons, right? And when it has the plus, that means it doesn't have the electron yet. It's when it's NADH, that's when it has the electron. So we need this molecule. We need NAD+ to strip away those electrons. And in the process that I just taught you, with cellular respiration, that's not a problem. Because the NAD+ grab the electrons, take them over here, dump them off to the electron transport chain. Now it's ready to grab more electrons. So it comes over here, grabs the electrons, shakes them over to the electron transport chain, and so on and so forth, right? The NAD+, when we're doing what I just taught you, which is regular cellular respiration, they had a place to dump off those electrons. But when you don't have oxygen, it doesn't have that place to dump off those electrons, right? So that gives it a new problem. So what happens is this NADH drops it back. No, excuse me. I don't want to tell you what happens yet. First, let's look at this picture. So again, this is what I just taught you, right? This is cellular respiration. And I put a big old X through this because this is what does not happen, right? If you don't have oxygen, these electrons have been stripped away by the NADH, or excuse me, NAD+. They have nowhere to go. So now we just have a bunch of NADHs hanging around. They can't accept electrons because they're already full. And if we can't strip away electrons, then we can't produce ATP. And if we don't produce ATP, then we die, right? So what's the solution to the problem? And the answer is recycling. Recycling NAD+ cannot occur in anaerobic conditions because, again, this is everything we said in writing. There's no oxygen to accept those electrons. So instead, and this is the part you need to know for the exam, again, like Kesterson brought this up before I even started talking about it, electrons have to come from somewhere. They have to go somewhere. So if there's no oxygen to accept those electrons, where do they go? They go to the pyruvate. So you do need to know that. When we're talking about cellular respiration, this is what we talked about previously. Oxygen accepts those electrons. Take away the oxygen, the electrons still have to go somewhere. So where do they go instead? They go to the pyruvate. And again, once that NADH drops off those electrons, it's technically called NAD+ again, and it's open. It's ready to accept more electrons. So again, we're still recycling the NAD+. But now, instead of the electrons going to the oxygen, they're going to the pyruvate. And this just keeps going, right? This keeps glycolysis working, right? Strip away the electrons, dump them somewhere. Strip away the electrons, dump them somewhere. And here's another thing you need to know for the exam is this bullet point right here. When we add those electrons to the pyruvate, we produce something called this, lactic acid. So there's two different types of fermentation. I'll go ahead and tell you now. So this is called lactic acid fermentation, and we'll talk about a different one later, but this is another thing you should know. When muscle cells, because remember that's what we're talking about mostly, when muscle cells go through fermentation, what do they produce? They produce lactic acid. You need to know that. But just so you know, and this part will not be on the exam, that lactic acid is then transported to the liver, and the liver converts it back to pyruvate, and then the pyruvate can go back and the whole process starts again. And again, you can see that will probably be, it's quite the process, right? The lactic acid having to go all the way to your liver, and then having to be converted back to pyruvate, and the pyruvate making itself back, making its way back to your cells. So again, this is why we can't survive without oxygen indefinitely. We can only do it for a little bit. So any questions about this slide? Before I show you a picture of this slide, yes.
Well, when we're dying, we're gonna have burning sensation.
That's a good question. I guess it depends on how you die. If you, I bet if you die due to a lack of oxygen, you would.
Yeah.
It's good.
You're gonna be without oxygen, you die.
Well, yeah, but I mean, if you just get your head blown off, you're not gonna feel anything, for example. All depends on how you die. If your brain dies, before your heart, for example. Great question. Also a good independent work topic, because I don't know that for sure. It just makes sense to me that if you're dying of a lack of oxygen, I imagine it would burn. I do know though, like for example, and that's not the same as being in a situation where you run out of oxygen necessarily. Like if you were stuck in a place where, oxygen couldn't come in, but also carbon dioxide couldn't go out. Eventually, I think you might die of carbon dioxide poisoning before lack of oxygen, but I don't know that for sure either. Very interesting to look at. I did look that up last summer with that sub. What's the name of that sub that went to the Titanic? Anyway, remember it was missing for hours before they found out it imploded. And I was wondering, man, what a horrible death. And I was looking that up, like, what would it be? How would the death be? Another independent work topic if you were to look into it. What happens if you're stuck in a vessel? You can't get any more oxygen in, you can't get any carbon dioxide out. Anyway, here's a picture of what I just told you. This is not your textbook, so again, if you want to study this picture, you need to download it. Again, this is fermentation in muscle cells, right? So this is happening on the glycolysis. We have the input of glucose. Just like usual, we're breaking glucose down to produce pyruvate. We're stripping away high energy electrons. In this case, the electrons don't have anywhere to go, or they don't have the oxygen to go to. So instead, they're added back to the pyruvate, and that produces lactic acid. So there we go. So that's where you might get the burn from, right? Because you're producing acid. You'd like to have acid in your muscles. And there's other ways, other things that are making it burn too, but yeah, that obviously has something to do with it. So like I said, this picture is not from your textbook. That's from the old textbook. Here's the picture from your textbook, your free textbook. Oh wait, no, I'm sorry. This is not a picture from the textbook. This is an animation. So if you download the PowerPoint, you can watch kind of everything I just said, but in action. Any questions about that?
All cells are able.
Oh. So here we go. That's one type of fermentation. There's another type. You need to know the difference between the two for the exam. The other type is fermentation in microorganisms. Your book didn't get specific about this and differentiating, but I want to. So again, what you produce is lactic acid. What microorganisms produce is not lactic acid. But here we go. Let's talk about this. Fermentation can sustain many microorganisms, right? So not us. Fermentation doesn't work for us. It won't keep us alive that long. But if you're a single-celled organism, for example, you don't need a lot of oxygen. So in that case, fermentation will work. And yeast is a good example. Yeast is one of those interesting organisms because it is single-celled like us, it is single-celled like a bacteria, but it's a eukaryotic cell like us. So it can do both. So really quickly, before we talk about the other type of fermentation, let's talk again about lactic acid. This is just an example. I'm not going to ask you anything about this, but I find it slightly interesting. Yeast can also make lactic acid, and that is used to make things like cheese, sour cream, yogurt, soy sauce, pickles, cabbage, olives, sausage, meat products, all that stuff. You'll look that up for independent work too. How is lactic acid from yeast used to make different food products? Not important for the exam, but it's there if you want to look into it. Soy sauce, all that stuff. But yeast can also make other stuff. Let's see how many of you, other than in this class, what do you think of when you think of yeast?
Bread.
Bread's a good one, okay. And that'll come up here in a second, sort of. What else?
Beer.
Alcohol, right? And that's the big one, alcohol. Yeast is also, first of all, yeast is capable of cellular respiration, like we just talked about earlier. So it is eukaryotic. They can do respiration just the way I taught you, glycolysis, citric acid cycle, electron transport chain. But it can also perform alcoholic fermentation. So if you take away the oxygen from yeast, obviously it's gonna have to do fermentation because there's no place to put the oxygen, right? So it has to do fermentation. But instead of producing lactic acid, well, in addition to producing lactic acid, it can also produce alcohol. So you need to know that, right? Well, yeast or the other type of fermentation, you don't necessarily need to remember that it's from yeast, can also produce carbon dioxide and alcohol. And technically, yes, it is ethyl alcohol, aka ethanol. Before the exam, I'm not going to get tricky with you and have you differentiate between that type of alcohol or any other alcohol. You should just think of fermentation. There's two types of fermentation. One of them produces lactic acid, and the other one produces alcohol. Just remember that. And also remember, technically, the one that produces alcohol also produces carbon dioxide. Which goes back to what you guys were saying when I asked, what do you think of yeast? A lot of you said bread, and that's how bread gets its fluffiness, right? Because it produces carbon dioxide, and that makes the bread rise. There's a picture of everything I just said. It looks very similar, right? It's glucose broken down into pyruvate. We're stripping away the high energy electrons. We're adding it back. But in this case, we're also breaking down the pyruvate some more, right? So those pyruvate broken down some more, and that's where we get the carbon dioxide, and then that's where we get the ethyl alcohol. And the last word for attendance will be alcohol. So I will see you guys on Wednesday, or I'll be online for office hours if you need me.